Okay, here's a quick topic right before we get to enzymes that I wanted to separate from the cell portion because this talks a bit about DNA. Differentiation is something that is a big deal in a multicellular organism. Without it, multicellular organisms can't be built. Turns out that environmental and cellular interactions with DNA in the nucleus causes cell specialization and the ability of cells to become different. So that a multicellular organism can have a liver and a kidney and a spleen and a pancreas and a stomach that all have the same chromosomes, but they're different from each other. Differentiation in development is due to external and internal cues that trigger different genes to do different things in different cells. We talked in an earlier chapter about homeotic genes and how important homeotic genes were to development. Furthermore, homeotic genes are generally not used at all in the adult phase of a multicellular organism, but they're used during embryonic development. Embryonic development is extremely complicated, so it requires a completely independent gene set to actually cause it to unfold properly to build a body. Once a body is built, the genes are shut down, and then housekeeping genes and differentiation genes are responsible for making the organism work. So in insects, a protein known as bicoid, if you remember, helps to determine the anterior and posterior axis. Bicoid is one of the homeotic genes of an insect. It helps the insect body divide into segments. Structural and functional divergence of cells in a multicellular organism during development is due to different genes turning on in different cells, which causes the cells to make different proteins and become different from one another. Simultaneously, those cells are suppressing other genes to prevent them from making other proteins, which would prevent them from differentiating. So different cells of different developing organs in an embryo express different genes. The differing genes produce different cell receptors that allow different cell lines to respond to signals differently. Environmental stimuli can affect gene expression in a mature cell. Pathogens routinely affect the gene expression of immune cells. So when you get infected with a virus or a bacteria, your immune cells actually experience specific types of gene expression that allow them then to manufacture lots of antibodies, which in turn causes the destruction of the pathogen. To do this, they undergo, if you remember, a clonal expansion. Immune cells undergo clonal expansion and they produce all kinds of different proteins in response to the pathogen so that they can end up controlling the infection. So, the white blood cells in the human body are, if I can fit that right there, they come from stem cells in the bone marrow. And in the bone marrow, a progenitor cell, depending on where it is in the bone marrow and what chemicals are stimulating it, it can produce a whole different lines of cells. Some of the cells that stem cells produce are granulocytes, which are the basic war machine of the human immune system. Some of them become monocytes, which are the they're superheroes compared to just ordinary soldiers. Some of the stem cells make T cells, which are the cells that control all of human immunity. Some of the stem cells become B cells. B cells make antibodies as far as the immune system is concerned. And some of the stem cells divide and become thrombocytes, which are in turn go on to become platelets. So the point here is that these cells can be made because this cell is being stimulated differently in different areas to produce these different cell lines. Once these cells have determined their fates, the granulocytes make slightly different proteins than monocytes, which make slightly different proteins than T lymphocytes, which make slightly different proteins than B cells, and so on, so that they can be different. Thrombocytes or platelets clot your cells. These make antibodies. These control all your immune cells, although they further differentiate into helper T cells, suppressor T cells, or activated or even killer T cells, which aren't shown in this diagram here. Monocytes go on to become macrophages, which eat massive amounts or prodigious amounts of foreign material, and granulocytes are your basic soldier of the immune system that eats non-specifically an awful lot of foreign cells that get into the body. This is what we call cell differentiation. This happens in an adult organism. Most cell differentiation happens as an embryo, but the immune system is a classic example of differentiation that happens even in an adult organism. White blood cell differentiation is absolutely critical for survival. As you've probably all heard of, AIDS is a disease where you are missing a specific type of white blood cell, and that is this one. And without this blood cell, you lack the helper T cells, these guys, that can actually stimulate, especially your macrophages and your B cells, to do their job. So without these cells, these two cells won't do their job because they can't be stimulated to do their jobs. That interaction is part of the differentiation process. So taking these cells out of commission, which is what the HIV or virus does, the AIDS virus does, it disables your immune system from the top down. Okay, so the next topic we talk about in here is enzymes, and I'll put that in a separate video.